In our last discussion, we established this idea that because most mutations that occur are effectively neutral, that we can use them as an estimate for how long it's been since two species diverged from one another. And this works generally on the principle of predictability of random events over long periods of time. So let me give you one more analogy. Let's say I asked you to estimate how many people had won the lottery over a certain period of time. Now, if you had enough data, if you knew what the odds were of winning the lottery, and if you knew how many tickets were sold on average each week, you could estimate over a substantial enough period of time, on average, how many lottery winners there were each week. And it might come out to less than one, it might come out to a small number. And so if I asked you to estimate how many are going to win next week, you don't have a very precise prediction. So if your estimate is 0.5 each week, right, then you don't know whether there's going to be one winner or no winners, or maybe even occasionally 2 or 3 winners for each individual week. However, over long periods of time, all that randomness evens itself out. So if you say there are on average 0.5 winners each week, and I say how many people are going to win over the next 100 weeks, then suddenly you could say, oh, it's going to be 50, give or take, but you'd be pretty close to 50 because some weeks you'd have more, some weeks you'd have less, but all of that randomness would average out. And so the molecular clock is very, very accurate over long periods of time because it relies on this same sort of thing, this randomness. And remember, the figure that we are interested in when we're looking at the molecular clock is the mutation rate. So we need able to either measure directly the mutation rate or accurately estimate the mutation rate. And if we can do that, then we can use that one simple value to estimate divergence time across different organisms. Okay, now, there are a few other things that play a role that are a part of this, and this gets a little bit more technical and complicated, but we're not going to get into that technicality. The Ney paper does a pretty good idea, a pretty good job of giving you some of that technical advanced material. And if you're interested, go ahead and read it. But just use the discussion here as a guideline to the level that you need to know. And for our purposes, we're not going to get too much into the derivation and the math of it, but you do need to understand the role that a couple of things play. Now, when we're estimating the molecular clock, we are assuming a few things. That the variance across any one site is going to there's going to be some variance. And we use a Poisson distribution to estimate the amount of variance that we expect to see. And there are different ones depending on whether everything is pretty much roughly the same and only a few vary widely, or whether there's a wide rate variation across organisms. And so we can use this to account for some of that variation that we would expect to see. Again, with a random process, you're going to have some sites that are more diverse than others. Now, remember that this is only applying to sites that are evolving neutrally. If we have other things going on, natural selection as being the big one, then we would expect to see a violation of this distribution. Okay, and so the dispersion index, which follows a Poisson distribution, is one of the things that's mathematically used. And if there's a set of sites or a number of sites that violate this Poisson distribution, either that is a sign for strong natural selection for that, or perhaps we need to look at a different type of distribution, which can occur sometimes to account for violations of that. Here is another thing that we can do to test the molecular clock, and this is called a relative rate test. Realize that every node on a phylogeny, and we did this and emphasized this pretty heavily in our past unit, but every node represents a common ancestor. So that node labeled with an X on this diagram represents the common ancestor of 1 and 2. But this node a little bit more deep in the phylogeny, this node, maybe we put a little Y by it. That is a common ancestor, but it's a common ancestor for multiple species. And because of that, and if we have an estimate for distance between different species, these branch lengths, right, which are estimates of how much genetic change there's been between species 1 and species 3. But if we have a molecular clock in effect, then we would expect the genetic distance between the human and the monkey to be the same as the genetic distance between the old world monkey and the new world monkey. So realize that if a molecular clock is into effect, all of the lines tracing to that same ancestor, those genetic distances should be equivalent. So the relative rate test then becomes a test for whether or not we can assume a molecular clock. If the distance between the human and the New World monkey is not significantly different than the distance between the Old World monkey and the New World monkey, then suddenly we have good indication that a molecular clock is in effect, that in essence, everything is evolving neutrally. And so we could test this. We could test different data sets or different partitions within a single data set. So here we're looking at synonymous sites that are in 9 nuclear genes. We do a little test, and then we determine whether or not that follows our prediction. We maybe even do a statistical test to determine. And the ones that are significantly or not significantly different from what we would expect to see under the relatives rate test, they are evolving neutrally. So synonymous sites seem to be evolving neutrally. Oh, cool. We could use that to estimate divergence time. This gene, the gamma globulin pseudogene, this is a gene that's no longer under strong natural selection. It's evolving neutrally. Here are 3 introns, however, that do not pass the test, 2 flanking regions of a gene. So we could maybe determine whether or not a data set or certain parts of a data set were evolving neutrally and therefore could be used to infer a molecular clock. Now, there are some additional considerations that are still being investigated, but that we might need to consider. Now, remember, we on the outset said that every time a mutation occurs, it's during meiosis for eukaryotic organisms, right? That's when we have potentially mutations being able to accumulate in populations. So, is the molecular clock following a generation time clock, or is it following an absolute time? Meaning, if I've got an organism whose generation is much faster, let's say an insect who has a generation every single year, would we expect the molecular clock in that organism to be the same as for a human, where generation time is more like, historically at least, has been more like 20 years, maybe even a little bit less? By the way, human generation time is increasing, right, as people more and more choose to delay their reproduction and have kids into their 30s and 40s. But historically, it was probably 20 years or so or less on average for human generation time, okay? But just by the nature of how they live their life, insects are always going to have a shorter generation time than humans. So should our molecular clock reflect generation time, or should it reflect absolute time, meaning mutations per year? Now, it turns out that when you look at this experimentally, that there's a little bit of a unexpected result. So many people suggested, no, mutations should be fewer between because of long generation times in humans versus, say, in insects, right? So 1 generation and a mutation, or on average how many mutations occur. And the only thing that would offset this would be if the mutation rate, so originally people said, oh, we need to change this mutation rate to something that is adjusted for generation time G. So they suggested a new value. And then we began to gather data and experimentally look at it, okay? So here, the big circles represent the generation time for, say, humans. The little circles represent the generation time for, say, an insect. And we might need to compensate for that. But as more and more data was gathered in the eukaryotic organisms, we realized that this didn't seem to hold true. That the molecular clock, when we could infer that it was valid, that we were looking at neutrally evolving portions of the genome, was pretty much the same for organisms with many mutations versus more organisms with slower reproduction and fewer, many generations, and versus organisms with slower reproduction and fewer generations. That indeed it followed an absolute time on how many mutations accumulate per year. And so people begin looking for an explanation for this. And it turns out that the data seem to support this idea of, well, the non-deleterious mutations are very roughly constant per calendar year, not per generation time. And so there are a couple of ways we can look at this. People thought that perhaps, and this might play a small role, particularly in particular comparisons, but people thought perhaps maybe it was this idea that organisms that reproduce quickly have, on average, larger population sizes. And this isn't always true, right? But certainly for humans, probably our effective population size was much smaller than, say, rodents, which have a larger range in many cases, and because they're small, many more of them can be supported in a same-sized environment. And so people thought perhaps maybe this population size would help to compensate. But remember, we kind of threw out population size as a big impact on this, because population size basically compensates or offsets, if you will, the number of mutations, or it's offset in the 2 different ways parts of that calculation. Okay, so it turns out that when we look at synonymous versus non-synonymous, this idea seems to be held up. That deleterious mutations, there does seem to be a distinct difference for fast generations, that these deleterious mutations accumulate and are present at a higher level than in slow generations. But when we go to these non-deleterious, it seems to just be absolute time, or calendar year rather. Okay, and again, there's still people trying to figure out all the details of why that occurs, but you should know that the molecular clock follows a calendar year or absolute time rather than a generation time. Okay. Another thing that people thought might offset it, but again, this has been largely disproven, is metabolic rate. That organisms that are homeotherms, that have a higher metabolic rate, on average maybe have a higher mutation rate. And they do certainly. And so maybe we would expect to see them have a faster running molecular clock because they have more mutations. But again, it turns out that this largely has been disproven. So metabolic rate has very little impact at all on the molecular clock. And again, DNA repair. And this might play a role, but realize that when we're measuring mutations, we're usually filtering out DNA repair mechanisms because we're not often gathering data in real time and looking at how mistakes are made in real time, like the polymerase actually copying the DNA. We're looking at it after the potential repair mechanisms have had a chance to catch mutations. And so really our mutation rate that we measure is not the raw mutation rate. It's really a mutation plus repair mechanisms rate. How many of those actually make it into the organisms? And so DNA repair really does play a very low role and little role. So even though these were all big factors originally, right? Generation time, metabolic rate, DNA repair, largely they have been discounted. And they don't play a major role in the molecular clock. And so molecular clock really does just come down to this base mutation rate. And it's a good estimation if we calibrate it correctly using the fossil record. It becomes a really good, although maybe not very precise, but a very accurate measure of divergence time across wide ranges of species. And so as a wrap up for this, we're just going to see a reminder. This is what we would probably see after mutation and genetic drift have both played roles in a wide range of mutations, which is, again, most of the mutations that occur because natural selection only has an impact on a very small number. Okay? And so we have this idea of nearly neutral theory, that the vast majority of mutations that accumulate in populations are neutral. A large percentage of them are negative, and then some are going to be positive, and natural selection will work to expand this. And then a very, very few of them are going to be negative because natural selection is going to remove them. I had to pull it out here because I had trouble seeing that very tiny sliver of them that are positive. And so it's this large, vast majority of mutations that are neutral or nearly neutral that we can use to infer molecular clocks and then all those other things that we talked about that come along with neutral theory. And so although it might not be 100% neutral theory, because we do have some other things, that the vast majority of the diversity we see in organisms seems to follow the patterns that we expect to see when mutations are evolving neutrally.